Okay, the other part of our lab is hearing. In the upcoming lecture on Tuesday, we're going to talk much more detail about hearing. For right now, I'm just going to give you some very basic background on hearing. So when we think of hearing, there's the outer ear. It's like your pinna. That's what you think of on the side of your head. Leading into like that ear canal. So that's where the vibration is coming into the little ear canal. This is supposed to be your skull here. Here's your brain. So the thing that is going to actually convert the vibration into an electrical signal, that's in what's called the inner ear. And it's located, don't worry about the detail of it right now. It includes the cochlea. So it's got this thing. So this part here that I've just drawn, this is my inner ear. Right, so this little thing here is going to send a message to the brain. So that's going to be something, again, we're going to look at in much more detail once we get to the ear specifically in lecture. But for right now, just think about this inner ear as being the place where you have the machinery that can convert vibration into electrical signal, which is going to create this vestibulocochlear nerve, which goes and sends the action potentials to your auditory cortex and everything. The other part of the ear is getting that vibration into the inner ear. And there's two parts to that. So the sound coming in, most people know about the eardrum, also called the tympanic membrane. So when the sound is coming in, the tympanic membrane is going to vibrate back and forth. It really is like a little drum head, right? If there's noise, if you just rest your hand on a drum and there's loud noises around, you'll actually feel that the drum head is vibrating sympathetically with just the noises in the room. And then we need to transmit that vibration into the inner ear. And there's this little system of levers. Like there's this thing called impedance matching. If you have vibrations in the air, but the little cells that convert the vibration into electrical signals, they're all in this watery environment. We need to convert the air vibration into like vibrations in the watery environment in the inner ear. So we don't want the energy just to bounce off. We want it to go and really maximally stimulate the inner ear. We have these things in the middle ear here. We have these little ossicles, the little bones. Little ear bones. The hammer, the anvil, and the stirrup. So they are basically like a little lever system that pushes and pulls on this little, the oval window that we'll talk about. But it's basically a way, as the eardrum goes in and out, these little ossicles, little ear bones, push and pull on this little, what we call the oval window here. The oval window is a little membrane that you can send the vibrations into the inner ear through. And then once the vibrations are in the inner ear, they get converted into an electrical signal, which then goes to the brain and you experience sound. Um, the only other thing I need to include in this picture is the fact that this middle ear and inner ear are all located within the skull bones. They're all within the temporal bone of your skull. So let me do that now. Um, so everything I've kind of drawn here is kind of grayish is the skull. And this is going to be important in a few moments when we talk about the different kinds of deafness. So a lot of our lab is going to be about looking at different kinds of deafness and the tests we have to test for deafness. So we should talk about the two main types of deafness, different ways that different things that can go wrong that mess with your ability to hear. You know, what's one way that is what can go wrong that would make it so you couldn't hear?
Something with the ear bones.
Yeah, if there's something that keeps the vibration from ever getting to the inner ear in the first place, that is going to make it so you can't hear. So the inner ear might be fine. The inner ear might have all the machinery to convert vibration to electrical signal, but if the vibration doesn't get there, that's going to be what we call conduction deafness. So you can have conduction deafness. So this is the vibration never reaches the inner ear. And again, you know, that can happen with a number of different ways. It could just be because you got way too much earwax and the sound isn't getting in. Or it could be you have a burst eardrum, so the eardrum isn't vibrating properly. Or maybe your ossicles are fused because of overgrowth of the bones or something. Or maybe you've got some ear infection and all this fluid filling up in here that and swelling that keeps things from vibrating properly. For some reason, whether or not it's a burst eardrum or the bones locked up, the vibration never makes it to the inner ear, so you don't hear it. That's conduction deafness. The conduction is not, you know, the path, the vibration is not being conducted to the inner ear. What's the other thing that can go wrong?
Could it be something with like the nerve?
Yeah, so the other thing is what happens if the inner ear or the brain is not working properly? Maybe the vibration is getting where it's supposed to go, but the machinery that converts vibration to electrical signal doesn't work. There's no sensory transduction. So that is called sensory neural deafness. Right? So the vibration is getting where it needs to go, but you are not able to actually convert that vibration into an appropriate electrical activity in your brain. Right? So sensory neural deafness, conduction deafness, they're very different, but the effect is the same. There's a sound and you don't hear it. Um, if you have sensory neural deafness, is there anything you could do to actually hear something, make a sound appear inside your head?
you wear hearing aids?
So a hearing aid wouldn't even help for that, unless you do like a cochlear implant, which we'll talk about in lecture next time. Like it is possible to do a really fancy system where you thread electrodes up into the cochlea and electrically stimulate parts of the inner ear. But in general, sensory neural deafness is done because the basic machinery that converts electric and vibration to electrical signals isn't working. Nothing's going to help. Conduction deafness, though, in conduction deafness, the problem is that you're just not getting the vibration to the inner ear, even though the inner ear is working. And what you can do is you can actually just vibrate the skull directly. So what we call bone conduction. Let me do a new sheet. And here's the brain. So if the sound coming in is blocked here, but this part is okay, what you can do, and this is called air conduction. Air conduction is if the vibration is coming through the air, vibrates the eardrum, and then continues in. But you can also, like, let's say, and this is usually done by putting like a tuning fork on the skull. Tuning fork is just this thing, you go bing. If you vibrate the skull itself directly, the inner ear is embedded inside the skull bones. So you can actually just vibrate the inner ear by vibrating the skull, by pushing something against the skull and vibrating the whole skull. You know, this is going to be part of the hearing tests. Like if somebody can't hear anything through the air, if I have my tuning fork, let's make it a color. If I have some tuning fork here and I hear nothing, but then I take the tuning fork and I push it against my skull bone, but I do hear something, that's important information. That says air conduction isn't working, but bone conduction is working. So what kind of deafness could I say is probably happening here?
Conduction deafness.
It's conduction deafness, right? Because I have evidence that the inner ear is working. If you directly vibrate the skull, I can hear something. The thing that's not working is that conduction to get there in the first place. So that is at the core of these hearing tests, is this idea that you have air conduction versus bone conduction, and that they can tease apart conduction versus sensory neural deafness. When you take the places that you put the tuning fork, if you're checking for bone conduction, sometimes you just put it like right on their forehead or on top of their head. Sometimes you put it here as well, which is the mastoid process. That kind of supposed to be a tuning fork, right? If you feel it's that bony bump right behind your ear, because that's directly stimulating the temporal bone. So that's another place where you can try to stimulate bone conduction. And again, as long as the inner ear is functioning, if they don't have sensory neural deafness, they will hear something with bone conduction, even if they don't hear it through the air. So that is, I think that's the background you need for that. I'm trying to think of the other things we need to talk about here. We should talk about this range of hearing. remember I've mentioned that with receptors, it's not just about the whether you can detect a certain modality. It also depends on different qualities, like the intensities you can detect, the frequencies you can detect. For a human, you know, maybe up to around 20 kilohertz or so. 20,000 cycles per second. Um, that's if you are like a kid. By the time you're in your 20s, it's maybe down, it's down below. Like by the time you end up in your 30s, it's even lower. I think right now I'm around 13 kilohertz. It goes down and down. As you get older, the hair cells that respond to the higher frequencies start dying off and they don't repair. So, do you know what was the word for old vision? The fact that your near point just got farther and farther away.
Presbyopia?
Yeah, presbyopia. So here we end up getting presbycusis, like old hearing. The older you get, the lower and lower the frequency is that you can actually hear. Right? There are actually convenience stores that will play sounds that are really high frequency like this because it's incredibly annoying to a teenager, but an adult that they are not trying to like push away doesn't hear anything. Um, the same ways people like kids have come up with like text chirp tones that they can hear in the middle of the class that their teachers can't hear. Um, Normally it's kind of fun to do this in class because we actually map it out and people can see like, well, I don't hear anything. And this person is like, turn it off. My ears hurt. Right? If you don't have the ability to hear a certain frequency, then it's just like silent to you. Kind of like, and different animals, like I said, cats, they go up to like over 50 kilohertz. Stuff that they hear, you don't hear at all. And bats, da 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 da. Bats can hear like up 100,000 hertz, 100 kilohertz for their echolocation and stuff. So different animals can hear very different maximal frequencies. What we are going to do is have you all, there's a cool little video that you'll play. Also, this will be something you'll do at home. And it basically just sweeps the frequency spectrum. And sometime, probably like at the end of lab today, I'll have you do that and just email me with your data. Like what was the highest frequency you could hear before you couldn't hear anything anymore? You have to use kind of good, put on little headphones. It's better than if you just try to do it on like a crappy little tinny speaker, it's not going to work as well. And then we will look at that. We will actually, just the same way that we kind of graphed age versus near point, part of your lab write-up is going to be age versus kind of what is the top frequency you can hear. And it's just normal. It's like, unfortunately, it's a normal thing that your vision gets worse, your hearing gets worse, your reaction times get worse. Everything goes downhill. One thing that's not so bad about this is most vocal stuff is in the range of 3 to 5 kilohertz. So you can have a lot, your hearing can be pretty compromised, but you can still understand voice and talking. You might not be as good at picking out one voice from another, and if it starts getting loud around you and stuff like that. when it gets really low, I tested my mom recently, and she literally couldn't hear above about 1.5 kilohertz. You know, you know, so it can get that's extreme. But there's for a lot of people, like when they even like they hear music, they don't hear any sizzle of the hi-hats and the cymbals. They just kind of it's just much more muted. The more and more you listen, the more, yeah. So I would say, public service announcement, earplugs, if loud, right? I don't know about you. When I was in college, you were a total loser if you put on earplugs at a ear splitting concert, and you don't want to be a loser, and instead you end up losing your hearing as you get older, and then you realize, oh, duh, I should have worn earplugs. So anyway, I would say, I think it's getting less of a thing. Because now I go to shows and lots of people put in earplugs, and there's actually good earplugs now, that are pretty cheap. That pretty cheap, like around 15-20 bucks, that will cut all the different frequencies kind of pretty similarly. Like the old school, if you just put in the little foam earplugs, they cut the high frequencies without the low frequencies, and everything sounds really muffled. So it's like, then I'm not really enjoying the music. But there are a lot of cool new kinds of earplugs that just cut the volume at all frequencies, so the music still sounds good. It just doesn't sound as loud. And then you can actually enjoy it more because you're not going to be in the back of your mind thinking that you're destroying your ears. So so just putting that out there. And it's not just me. There's a lot of kind of musicians and such out there that talk about this, that feel, regret that they messed up their hearing by not taking care of, um, exposure to really loud sounds. So there's frequency range. Oh, the last thing I'll talk about, and then we'll just go into the actual procedures, is localization. So if you're sitting here, with your eyes closed, there's a blindfold on, and there's some, I'm trying to, God, I'm trying to do like a snap. Snap. This person can probably say, oh, the snap came from that side versus the other side. So we're going to talk about what are the cues, what are the information that this person's nervous system, brain has to figure out that the snap came from there versus the other side. Any thoughts?
You hear it more in one ear than the other? Like the vibration's higher in one or stronger in one ear than the other?
Yeah, so one big thing is it's going to be louder in the ear that it's closer to. You've got two ears, which is going to make this a lot easier. So we have what are called interaural intensity differences. Interaural means between the two ears. So intraoral, between the two ears. Intensity, just like the volume. Right? So the ear that hears it louder is probably closer to the sound source. So that should feel pretty intuitive. That's going to be one way that you can detect or can determine where it's coming from. What's another thing that's also different between the two ears? if you snap on one side versus the other. So it's important to realize, maybe if you haven't thought about sound, has anybody ever like seen lightning and then like 5 seconds later you hear the thunder?
So it'd be like the timing.
The timing. Sound is really slow. Sound is only like a few hundred meters a second. It's compared to other, it's actually, even though I talk about your neurons being super pokey, they're actually fast enough to determine what we call the interaural time difference. Right, so this is this other thing. One is we have the intensity difference. How is the volume different between the two ears? The second thing is the timing. If it reaches this ear before it reaches that ear, it's got to be on this side compared to the other side. So we have interaural timing differences and interaural intensity differences. And those, along with some other things, there's other things that, given the time, I don't want to get into. You actually have a few other things, even the shape of your ears, the fact that your ears are not symmetrical front to back, and top to bottom, that actually helps you because it changes what the sound sounds like if it's coming from one direction or another. Where do you think you're going to have the hardest time localizing sounds, given what we've just been talking about here?
Behind you.
Coming from behind you.
Behind, why is that?
Because the way the ears are shaped, the pinna kind of comes out, so it has to like get around the pinna to get into the hole.
No, so that's not going to be so much of an issue.
Because it's coming from the back, so everything's kind of equal.
Yeah, and it's not just the back that is going to be a problem. So going back to what Grace is saying, where else is it equal?
In front of you,
In front of you. And where else is it equal?
above you.
Above you. Anything that's on the midline, right? Anything that's directly on that midline, where the volume and timing are going to be identical to each ear, is gonna be trickier. Like I said, there's other things that you can use, like the shape of the ears, and actually even your brain adapts very quickly what you're wearing. When you change clothes, you actually remap your processing of sound to adjust for how the clothes you're wearing are absorbing the sound and how that affects how it sounds from one direction or another and stuff. But in general, if you are, in fact, this is something I also want you to do at home and to record in your lab notebook, is you are going to have your subject and they're going to just like, they can be standing, or you can, maybe it's having sitting is easier because that way you can get above them without, this just looks horrible. So they're sitting here, and you, in lab, we often use like, this guy looks like he's about to get electrocuted for, yikes. Make him smiling so he's not about to be executed. All right. The snap. Snap to the left. Snap to the right. Snap in front. Snap behind. Snap above. And record in your lab notebook. How well did they localize? Again, their eyes are closed. And they have to just point. It's over there. It's there, or it's there. And so write in your lab notebook, how accurate they were in pointing to the sound source. And then you'll interpret that. But think about like what Grace was saying, this idea of things on the midline where the time and the volume to both ears is exactly the same since everything's equidistant. That's probably going to be trickier than things that are more lateralized. When you're moving around the person, make sure that you don't shuffle so they can track you where you're moving or walking or something. So be kind of a hobbit, like just before, they have no idea where you are, and then they just hear a snap. So, and again, just try the left, try the right, try above, try in front, try in back. Those are like the three, more than three, 1, 2, 3, 4, 5 main things you should check. And record and see how it matches what we've just been talking about. And then we can share this on Monday as well. Not Monday, on Tuesday after a break to see what people found. But in general, we do find what we've just been saying, that there is a pretty clear degradation in your ability to localize if it's like on the midline. You might kind of know, you might point in front when it's right above you, or above you when it's right behind you, or something like that. So you'll get to see that. All right. So I am going to share screen. So in terms of the things to do for this hearing lab, doing the hearing lab. The first part, sound localization. So just what I was just saying. Again, and this is only going to take you like, 5 minutes. You're going to have somebody in it's probably going to be kind of fun. You have them sit down, make a little chart maybe ahead of time for like right, left, above, behind, in front of. So you kind of you're ready to write down what you find. Makes it easier. And, snap your fingers and get a sense of, what's going on. And again, there's like a little.
In this lecture, we're going to learn about way of saying where it
and all the stuff I've been talking about, you can watch her and she'll talk about it as well. But it's just kind of a review of what I just talked about. But when you're thinking about it, it's often useful to have somebody else talking about it too. Frequency range of hearing. So part A, it says ignore this part. Normally we have people like try different frequencies, but we're not going to do that. Then it says, using the videos below, determine the maximum frequency you can perceive and record that value. I say, please email me with your value and your age. I will take this and provide a de-identified graph of our class data for you to use. And these, we won't do this over Zoom because I don't know, it's probably going to degrade the sound quality and stuff.
This is Joe itself.
But
today we're going to try to find out how.
Basically, this one here, right? So don't worry about the years old. That's kind of stupid. What you want to worry is the frequency in hertz. We're in a science class here. So follow this and see what frequency does it say when you start hearing something. And like right now, because it's playing weird, I actually hear weird clicking, even though I know I can't hear at this frequency. It should sound like some really crazy, and it's going to keep going. And at some level, you're going to start hearing something like all of a sudden I just heard like a piercing screech in my ears. It was a little above this, but right. So don't do it through Zoom. Zoom has all these things that try to optimize for voice, but then it messes with what sounds sound like. So do this at home. Earphones are best. And note the frequency where you hear it. This other one here.
As we grow older, we often lose the extreme ends of our hearing spectrum. So, how many of the following sounds can you hear?
You know, so again,
Prepared. But let's keep raising the.
you know, so again, you can use both the videos to double check that you got a good number. So that's going to be another thing. It should only take you like 5 minutes. You just go through these videos and listen and find what frequency. Again, normally if we were in lab, I'd be playing these out loud and people would raise their hands and write stuff down. But you can't do this over Zoom because you need better sound quality. Just as a teaser, like this was a hearing from like a few years ago. You know, you usually get pretty clear negative correlations as presbycusis. Like people who are 20 are hearing up around 18, 19 kilohertz. People who are getting to be 30 can get up into 15. People who are up in their 40s and 50s are, you know, down here. And this is normal. Again, this is not like necessarily about hearing damage. This is just normal. Your ears lose the ability to detect high pitches as you get older. But you know, sometime over the next few days, if you could do this and email me, and then I'll email everybody the final spreadsheet of our class data. So I'll need your age and your maximum frequency that you can hear. Then the last ones are the RINNE and Weber tests. So these are the ones that use the bone and air conduction to determine your hearing. And again, normally we would do this in class, but instead you are going to watch videos of people being examined. And I'm going to describe this a little more in a few moments, but basically these first two videos just kind of describe more how the tests are done. They're also described in your lab book. And then it says here, watch the following videos. Evaluate the results for patients 1, 2, and 3. Record in your lab book. Be sure to interpret. So here you have patient 1, and she's doing the RINNE test and the Weber test. You're going to record the results for that patient and then interpret what the meaning is. Patient 2, they're going to give them a RINNE test and a just going to take a look at both the RINNE and the Weber in the same video. The same thing here, RINNE and Weber test in the same video for patient 3. You know, write down what the results are and interpret it. I can talk a little briefly about Oh, I need to stop this. So the RINNE test is very similar to what I was just saying in terms of comparing air to bone conduction. You know, in general, if somebody's ears are working well, air conduction should be much more sensitive than bone conduction. So what you do is you take this person, you put the tuning fork on their mastoid usually. You know, the tines, the little vibrating parts of the fork are pretty far away from the ear, so they can't really hear the vibration through the air because that's far away, but they do hear the vibration through the bone here. You know, and you start a timer. Tick, 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 tick. And you see how many seconds can they hear it before they don't hear it anymore. It might be 30 seconds or something. You know, eventually this starts vibrating less and less. The volume goes down. At some point, they don't hear it. The moment they don't hear it anymore, you shift this and put it right in front of their ear. So now we have air conduction going in. And all of a sudden, they should hear it again. Because even though this is vibrating not so much anymore, it's still plenty enough to do the air conduction and trigger the ear through air conduction if the ear is happy and healthy. And you should be able to continue to hear this thing vibrate for just as long as you heard it for the first part when it was the bone conduction. Right? If you stop hearing the bone conduction and you shift the tuning fork around to air conduction and you don't hear anything, that's a bad sign. Because that means that the air conduction is not working properly. Right? That's going to be a sign of what kind of deafness? I can't hear anybody.
Conduction?
Yeah, it'd be conduction deafness. What if I stuck this thing on the mastoid and I didn't hear anything, even with this vibrating thing against my bone? What would that mean?
Sensory neural?
It'd be sensory neural because you're vibrating the whole inner ear and you're still not hearing anything. That means there's something deeper wrong. It's not just about the vibration not getting there. It's about the system not being able to transduce the sounds. So that's the RINNE test. This one is, I think, more intuitive to me. The other one, the Weber test. It's kind of, think of it kind of like the unicorn test or something. You basically stick the tuning fork right on the center line of their head. It can be on the top of their head, it can be on their forehead, doesn't really matter. If everything is working right, and then you listen to the volume in the two ears. How should the volume compare in the right and left ear if this person is got good hearing, normal hearing? If you're right on the center line, assuming that you're not like asymmetrically deformed or something and each ear is like kind of symmetrical on either side of that center line, what do you expect to hear if you compare the two ears?
Equal?
Equal. It should be equal. You shouldn't hear any volume difference between the two. So if you hear it the same in both ears, it would mean that your hearing is normal. Or the other thing it could mean is if you have hearing loss, it's exactly the same on both sides. Right? So it can't actually tell you for sure it's normal, but it can say it's either normal or if you do have hearing loss, it's exactly the same on the two sides. What happens if it's louder? This is where it gets a little fascinating. Let's say it was louder on the right side than the left side. What would that mean?
You have a loss of hearing in your left ear?
So it could mean sensory neural deafness in the left, but it's kind of counterintuitive. It could also be conduction deafness on the ipsilateral side. And you should prove this. Like if you like hum, if you go like hum and then block an ear, block an ear and just talk. Right now, block an ear and talk. And do you notice that the ear that's clogged up actually sounds a lot louder than your other ear? Right? So again, it's a little weird at first, but once you actually do it on yourself, you realize it's true. That the side that has conduction deafness, where there's a blockage there, right now I've got conduction deafness if I'm blocking my ear, it's actually louder in the ear with conduction deafness. It's not exactly clear exactly why that works. It might be things like reverberate in there more and can't get out or something. So I can't tell you exactly why it's true. I can just tell you it is true. So with the Weber test, if it's the same in both ears, then we have a sense of it's all good or equal amounts of loss. If we have it louder in one ear, I have two options here: sensory neural loss in the opposite side or conduction deafness on the same side. So what am I going to do? How am I going to try to figure out the difference? Which it is? Do we have any techniques to determine the difference between sensory neural and conduction deafness?
The tuning fork?
But in particular, what would we do with it?
Put it on the mastoid process on the right ear, see if it...
Yeah, exactly. We go to the RINNE test, right? The RINNE test is the thing that's going to be able to distinguish between sensory neural and conduction deafness, right? Because then we can look on that particular ear, right and left ear. We'll do this on both the right and the left. We can say, aha, they don't hear it when it's there. So that's actually probably sensory neural deafness, but they do hear it, but they don't hear it with the tuning fork. So we know that this is the ear that has conduction deafness. Right, so between the two, that's why each of these things that I have you looking at are both RINNE test and Weber test. Between the two tests, you got enough information to make a pretty good diagnosis of what's going on with this person's hearing. So this will take a little bit of time, and what you just in your lab notebook, you'll write down like patient 1. Write down what you find for the RINNE test and Weber test. These are really freaking short. These are like a minute and 5 seconds, right? This isn't like, you don't have to like carve out a whole like half a day to do this. These are like 1-minute videos. You will write down what the RINNE test results are, what the Weber test results are. This is for patient 1. And then interpret the results. What is your diagnosis? And then again, these patient 2 and patient 3, again, these are also a minute and like less than a minute and a half each. And you just kind of record the results, what it means. And that is that. So are there any questions about any of these? No? It's so quiet. So the things that you have to do at home is the thing with the food cubes. That's probably going to take you the longest of any of them because you have to kind of prepare the little food cubes. But I genuinely think you're going to have fun with it. Do the thing with the sound localization, snapping around your subject. Get a sense of how we use two ears, both with intensity and timing differences, and how that kind of starts breaking down once you're on the midline. Do the thing with the frequency, the top frequency you can hear. And again, I'll share that class data with you all once we get that. And then, just do these little things with the Weber and RINNE test and kind of see how that works. And it'll also, by the time you're done with that, you should have a pretty good understanding of what is conduction deafness versus sensory neural deafness and how you distinguish them. What else? The labs are done then until, so next Tuesday, we're going to have just kind of a review for lab. So as you're writing everything up, kind of write down questions, make sure if anything that's not clear, we're going to have a day where we kind of go over this stuff, double check, look at, we'll look at, share people's results for the stuff that we're talking about today, make sure everybody is clear about everything. The lab exam, which is going to be next Thursday, is going to cover all the labs from the chemistry review lab up until this. It is open lab notebook, which means you can have your lab notebook there, but you can't have just random notes. Remember, your lab notebook should not be a place where you have crib notes and things. It should just be a record of your data, your analysis, your discussions, the procedures and things like that. But you will need to have your lab notebook. I might ask you what you found on a particular lab, but it's also just a really great resource because you are going to be able to, go back and it's like, what did we do? What was in this test tube and red bloods? Why did this explode? Oh, in my discussion, I wrote it up really clearly about how this penetrating solute did blah, blah, blah. It's like, thank God I wrote this up clearly because now I know what to write on my exam, right? So, that's why having a good, clear discussion and laying things out is really gonna help you dramatically in just having an easy time on this lab exam.
So the questions you were just saying, are those kind of similar to what we would see on the lab exam?
Yeah, or the lab exam is more likely to actually have pictures, like here's a picture of a test tube, what happened in it. So it's going to have some questions that are more just like multiple choicey kind of things, but because it's, if this was a real, like a regular semester, you would walk up to a lab station with stuff in front of you, kind of like an anatomy lab practical. You might have graphs where you have to interpret what you're seeing here, like what's going on in this graph.
Is the lab test going to be a lot of multiple choice or short answer or?
Yeah, I think you can think about the first exam, kind of a mix of those two. Like multiple choice are way easier to grade because they're graded automatically. But at the same time, I don't trust them totally. So I always make sure I mix it up. What else? Again, remember we have spring break. I don't know, here out in the desert, if you have your Seder, hope you have a happy Pesach and all that.